The field of biomedicine relies heavily on the scientific method and critical thinking skills. Researchers in this field must be able to formulate hypotheses, design experiments, collect and analyze data, and draw sound conclusions. This rigorous process is essential for advancing our scientific understanding and ultimately improving human health. Biomedical research requires a deep commitment to the principles of the scientific method, as well as the ability to think critically about findings and their implications. The work in this field has far-reaching impacts, which is why mastering these core skills is so crucial for success. Critical thinking is the ability to analyze information objectively, consider different perspectives, and make sound judgments. It involves asking probing questions, identifying biases, and seeking evidence to support conclusions. Critical thinkers don't accept information at face value. They challenge their own assumptions and the assumptions of others. They explore alternative explanations and seek to uncover hidden biases or flaws in reasoning. Evaluating evidence is key. Critically thinking requires carefully assessing the credibility and reliability of information sources, data, and research findings. This involves scrutinizing the methodology, identifying potential limitations, and drawing logical conclusions based on the available evidence. Critical thinking involves an analytical and evaluative approach to information. It requires actively questioning assumptions, considering multiple perspectives, and logically assessing evidence before reaching conclusions. Critical thinkers are skilled at identifying problems, breaking them down into manageable parts, and systematically exploring solutions. They are adept at weighing the pros and cons of different approaches. Critical thinkers are naturally curious and open-minded. They are driven to continuously learn, explore new ideas, and challenge their own beliefs and biases. This mindset is essential for advancing knowledge in the biomedical field. Critical thinkers approach problems with a spirit of curiosity, always seeking to learn more and understand the underlying issues. They strive to evaluate information and arguments impartially, setting aside personal biases and preconceptions. Critical thinkers ask probing questions, challenge assumptions, and delve deeper to uncover underlying principles and hidden complexities. They approach problems systematically, breaking them down into manageable components and exploring multiple solutions before reaching a conclusion. Critical thinking is essential in biomedicine, where healthcare professionals must make complex decisions that impact patient outcomes. By applying rigorous analytical skills, biomedicine experts can evaluate scientific evidence, identify biases, and formulate effective treatment strategies. This approach enhances patient care and drives innovation and advances in medical knowledge. Critical thinkers in biomedicine can scrutinize study designs, assess the validity of data, and identify potential limitations or confounding factors. This ensures treatment decisions are based on the strongest available evidence. Critical thinking enables biomedicine professionals to carefully consider multiple perspectives, weigh risks and benefits, and make informed decisions prioritizing patient well-being. By challenging existing paradigms and exploring new approaches, critical thinkers in biomedicine can drive breakthroughs in medical research, leading to improved diagnostics, more effective therapies, and better patient outcomes. The scientific method is a structured approach used by researchers to study and understand the natural world. It begins with careful observation of a phenomenon or problem that the researcher wants to investigate further. Based on these observations, the researcher formulates a testable hypothesis, an educated guess about the underlying causes or relationships. The researcher then designs experiments to test their hypothesis, controlling variables and ensuring the validity of their results. The experimental design involves determining the appropriate methods, tools, and data collection techniques to gather the necessary evidence. Throughout this process, the researcher collects and analyzes data to either support or refute their initial hypothesis. The scientific process starts with observing the world around us and the problems we want to investigate. We carefully examine the phenomenon, looking for any patterns or recurring trends in our observations. Based on these observations, we then propose a hypothesis, a testable explanation for what we've observed. Formulating a good hypothesis is a crucial step as it guides the design of our experiments and data collection. The hypothesis should be specific and based on our initial observations, setting us up for the next steps in the scientific method. The key elements of effective experimental design in biomedical research are a clear hypothesis, identification of variables, appropriate methodology, and incorporation of controls. Begin by formulating a testable hypothesis that directly addresses your research question and predicted outcome. Carefully define the independent, dependent, and control variables that will be measured and manipulated in your study. Select an experimental approach and techniques that are well suited to answering your research question. Incorporate necessary control groups to isolate the effects of your independent variable and rule out alternative explanations. Effective experimental design is crucial for ensuring the validity and reliability of your research findings. The key focus of this slide is on the important steps involved in the data collection and analysis process. First, we discuss the critical step of observation and measurement. This is where we gather relevant data through careful observation and precise measurement, such as collecting samples, recording experimental conditions, or tracking patient outcomes. Next, we cover the importance of data organization. This involves structuring and systematizing the collected data to ensure it is accessible and ready for analysis. This may include creating databases, spreadsheets, or other data management tools. Finally, we highlight the statistical analysis phase, where we apply appropriate methods to analyze the data, such as calculating means, standard deviations, or performing hypothesis tests. This helps us identify patterns, trends, and significance in the findings. Throughout this process, it's essential to maintain rigor, attention to detail, and a systematic approach to ensure the integrity and reliability of the data. The key steps in interpreting research results are identifying patterns, evaluating statistical significance, and considering biological relevance. When looking at the data, try to spot any trends or relationships that emerge. This can reveal important insights. It's crucial to determine whether the findings are statistically meaningful and not just due to chance. Careful statistical analysis is essential. Finally, assess the real-world implications of the results. How might these findings impact human health and disease? By taking this thoughtful, evidence-based approach, we can draw reliable conclusions from the research and lay the groundwork for further investigation or clinical applications. Peer review is a critical process where independent experts evaluate the methodology, analysis, and conclusions of research before it can be published. Replicating research findings is essential to validate the original work and build confidence in the reliability of the results. When replication studies produce contradictory findings, the scientific community investigates the reasons for the discrepancies, such as re-examining the original study design or exploring potential sources of bias. Addressing inconsistencies in research is important to resolve uncertainties and strengthen the overall scientific understanding. The scientific method, while powerful, has limitations that we must acknowledge. Our current understanding of the world can be incomplete or biased, leading to unexpected findings that fall outside existing theories. We must remain open to continuous learning and refinement of scientific models. Measurement errors, such as inaccuracies in data collection or instrument calibration, can skew the interpretation of findings. Accounting for potential sources of error is crucial. Replicating experiments and validating findings is a cornerstone of science, but factors like environmental conditions, sample variability, and uncontrolled variables can make it challenging to consistently reproduce results, limiting the generalizability of conclusions. Acknowledging these limitations is important for maintaining a healthy skepticism and driving further scientific progress. Confirmation bias. Researchers often have a tendency to seek out and interpret information in a way that confirms their existing beliefs or hypotheses. This can lead to overlooking or dismissing evidence that contradicts their preconceptions, skewing the results of a study. Selection bias. The way participants are selected for a study can introduce bias if the sample is not representative of the broader population. This can occur when certain groups are over- or underrepresented, leading to findings that do not accurately reflect the true population. Publication bias. There is a tendency for journals to publish studies with positive, statistically significant results more frequently than those with negative or inconclusive findings. This can create an imbalance in the available scientific literature, potentially skewing the overall understanding of a topic. Funding bias. The source of funding for a study can influence the research questions asked. the methods used, and the interpretation of results. Researchers may feel pressure to produce findings that align with the interests of their funders, leading to biased outcomes. Confirmation bias is the human tendency to seek out and focus on information that confirms our existing beliefs or hypotheses, while often overlooking or dismissing contradictory evidence. This selective information processing can reinforce our existing views, even in the face of disconfirming information. In the field of biomedicine, confirmation bias can compromise the objectivity and rigor of scientific research, as researchers may be biased towards findings that support their hypotheses, leading to inaccurate conclusions and suboptimal treatment strategies. It's important to be aware of this cognitive bias and make a conscious effort to consider alternative perspectives and seek out disconfirming evidence when conducting research or making important decisions. Selection bias is a common issue in research where the sample of participants is not representative of the broader population. This can happen due to factors like self-selection, accessibility, or convenience sampling. When the sample is skewed, it can lead to distorted or inaccurate findings that don't reflect the true characteristics of the population. In biomedical research, selection bias can significantly impact data on disease prevalence, treatment efficacy, and other critical metrics. This underscores the importance of using robust sampling techniques to ensure the study participants are truly representative. Researchers must be vigilant in their study design to avoid these pitfalls and draw reliable conclusions. Publication bias is the tendency for researchers and journals to preferentially publish studies with positive or statistically significant results, while negative or inconclusive findings often go unpublished. This bias can distort the scientific literature, leading to an overestimation of treatment effects and a skewed understanding of the true state of knowledge in a field. Techniques like funnel plots and Egger's regression can help detect the presence of publication bias in meta-analyses and systematic reviews. Understanding and addressing publication bias is critical to ensuring the integrity and accuracy of the scientific literature, especially in fields like biomedicine where treatment decisions can have major impacts on human health. Biomedical research must be approached with a critical eye to identify and address potential biases. Researchers should carefully examine study design, methodology, data analysis, and the validity of conclusions to uncover any biases. Assembling diverse research teams with varied backgrounds, perspectives, and expertise can help mitigate the impact of individual biases. Fostering a culture of open dialogue and balanced decision-making within research teams is key. Implementing rigorous study designs, such as randomized controlled trials and systematic reviews, can minimize the influence of biases and provide higher quality, more reliable evidence. Evidence-based medicine is an approach that emphasizes using the best scientific evidence to guide clinical decisions. It involves integrating clinical expertise, patient values, and the latest research data to provide high-quality care. Systematic reviews and meta-analyses are a cornerstone of evidence-based medicine. These synthesize results from multiple studies on a topic to provide a comprehensive, unbiased summary of the best available evidence. This helps support clinical decision-making by identifying the most reliable and up-to-date information. Systematic reviews and meta-analyses are powerful tools in evidence-based medicine. They systematically search for, critically appraise, and synthesize all the available evidence on a specific clinical question. This rigorous process helps overcome the limitations of individual studies and provides a more comprehensive understanding of a topic. By aggregating data from multiple high-quality studies, meta-analyses can generate more reliable estimates of treatment effects, identify sources of heterogeneity, and detect publication biases. This helps clinicians make more informed decisions and develop better-informed guidelines for patient care. Randomized controlled trials are the gold standard in clinical research. Participants are randomly assigned to either a treatment or control group. This helps minimize bias and establish causality between the intervention and observed outcomes. Observational studies analyze the relationship between variables without actively intervening. These studies can provide valuable insights into disease patterns, risk factors, and real-world effectiveness of treatments. Clinical trials in biomedicine often follow a phased approach. Start with small-scale safety studies, phase 1, then progress to larger trials evaluating efficacy, Phase 2 and 3. Finally, seek regulatory approval, Phase 4. Scientific evidence can be classified into different levels based on the quality and strength of the study design. The highest level of evidence typically comes from systematic reviews, meta-analyses, and well-designed randomized controlled trials. To evaluate the validity and reliability of scientific research, it is important to critically appraise the study design, methodology, and data analysis. This involves assessing factors such as the sample size, control groups, confounding variables, and statistical significance. Understanding the limitations of the study and the potential sources of bias is crucial when interpreting scientific findings. Considering the overall body of evidence, rather than relying on a single study, can provide a more comprehensive understanding of a research topic. Ultimately, the goal of evaluating scientific evidence is to apply it effectively in clinical practice, public health interventions, and policy decisions. Critical thinking skills are essential in this process to ensure that the evidence is relevant, applicable, and aligned with the specific needs and context. The hierarchy of evidence is a well-established framework for evaluating the reliability and strength of different study designs in scientific research. At the top of the hierarchy are systematic reviews and meta-analyses, which synthesize data from multiple high-quality studies to provide the strongest level of evidence. Randomized controlled trials, which use a rigorous experimental design to minimize bias, are considered the gold standard for evaluating the effectiveness of interventions or treatments. Observational studies, such as cohort studies and case control studies, provide valuable insights but are generally considered lower in the hierarchy compared to experimental studies. Understanding this hierarchy is crucial for clinicians and policymakers to make informed decisions based on the quality of available evidence. The goal of critical appraisal is to carefully evaluate the quality and validity of research studies. We'll start by examining the research methodology, looking at the study design, sample selection, data collection, and statistical analysis to identify any potential flaws or limitations. Next, we'll review how the study results are reported, including the clarity of presentation, interpretation of data, and consideration of alternative explanations. We'll assess whether the conclusions are justified by the evidence. Finally, we'll evaluate the overall quality of the research, considering factors like alignment with ethical principles, potential for bias, and generalizability of the findings. This will help determine our level of confidence in the study's conclusions. The goal is to develop a critical, analytical perspective when examining research to ensure we can trust and apply the findings appropriately. The goal of critical appraisal is to carefully evaluate the quality and validity of research studies. We'll start by examining the research methodology, looking at the study design, sample selection, data collection, and statistical analysis to identify any potential flaws or limitations. Next, we'll review how the study results are reported, including the clarity of presentation, interpretation of data, and consideration of alternative explanations. We'll assess whether the conclusions are justified by the evidence. Finally, we'll evaluate the overall quality of the research, considering factors like alignment with ethical principles, potential for bias, and generalizability of the findings. This will help determine our level of confidence in the study's conclusions. The goal is to develop a critical, analytical perspective when examining research to ensure we can trust and apply the findings appropriately. The goal of this slide is to outline the key steps in the case study and problem-solving process in biomedical research. First, researchers must carefully observe the patient's symptoms and medical history to formulate an initial hypothesis about the underlying cause of the condition. Next, the research team designs and conducts appropriate tests and experiments to gather relevant data that can help verify or refute the initial hypothesis. Finally, the team analyzes and interprets the findings, considering alternative explanations and looking for patterns, in order to draw conclusions about the most likely diagnosis or mechanism. This systematic approach is critical for advancing our understanding of complex medical conditions through case study research. Informed consent is a fundamental ethical principle in biomedical research. Researchers must ensure participants fully understand the risks, benefits, and purpose of the study before providing voluntary consent. When animal subjects are involved, Researchers have an ethical obligation to minimize suffering and adhere to strict protocols for their humane treatment and care. This includes using the smallest number of animals necessary and providing appropriate housing and environmental enrichment. Biomedical research participants should be selected fairly, without discrimination or exploitation of vulnerable populations. Researchers must consider the just distribution of the risks and benefits of the study across all participants. Researchers have a duty to uphold the highest standards of scientific integrity, including honesty, objectivity, and transparency in their work. This helps maintain public trust and ensures the validity of biomedical findings. Informed consent is a critical part of biomedical research and clinical practice. Patients have the right to be fully informed about the risks, benefits, and alternatives of any medical intervention or research they may participate in. Physicians must ensure patients understand and voluntarily agree to the proposed course of action. Patient autonomy is the fundamental principle that patients have the right to make their own informed decisions about their healthcare. Respecting patient autonomy means empowering patients to exercise their right to self-determination, free from coercion or undue influence. This is essential for maintaining trust and ethical standards in biomedicine. Informed consent and patient autonomy are best achieved through a collaborative, shared decision-making process between the healthcare provider and the patient. This approach encourages open communication, the exchange of information, and mutual consideration of the patient's values and preferences in determining the most appropriate course of care. Animal research is a critical part of biomedical advancement, but we must prioritize the ethical and humane treatment of the animals involved. Minimizing pain, distress, and unnecessary suffering is of the utmost importance. We have a moral obligation to ensure the animals are treated compassionately. Rigorous protocols and procedures must be in place to regulate and oversee the use of animals, adhering to all relevant guidelines and regulations. Institutional animal care and use committees provide crucial oversight, reviewing and approving all animal research proposals to uphold the highest ethical standards. By prioritizing animal welfare and research ethics, we can advance scientific knowledge while upholding our responsibility to treat all sentient beings with care and respect. Responsible conduct of research is guided by key ethical principles like honesty, integrity, respect, beneficence, and justice. These principles help ensure research is carried out to the highest ethical standards. Biomedical researchers must comply with a range of regulations and guidelines to protect the safety and rights of research participants. This includes oversight from institutional review boards, funding agencies, and governing bodies. Researchers have an ethical obligation to report any suspected cases of research misconduct, such as data fabrication, falsification, or plagiarism. Responsible reporting helps maintain the integrity of the scientific process. As researchers, it's crucial that we communicate our scientific findings clearly and effectively. This means writing in a way that is concise, structured, and easy for our audience to understand. Visual aids like figures, graphs, and diagrams can really help bring our research to life and complement the written narrative. But we also need to tailor our communication style to the specific audience, whether that's a scientific journal, conference, policymakers, or the general public. Ultimately, the goal is to ensure our research findings are translated into real-world benefits for human health. By communicating well, we can help bridge the gap between the lab and the bedside. The goal of effective scientific communication is to present your work in a clear, concise, and accessible way. Use simple, jargon-free language that can be understood by a broad audience, not just experts in your field. Well-structured writing with a logical flow helps your audience follow and understand your key points. Visuals like graphs, figures, and diagrams are essential for communicating scientific findings effectively. Make sure your visuals are visually appealing, easy to understand, and directly support the written content. When presenting, use a confident and engaging delivery style to capture the audience's attention. Highlight the key takeaways, use storytelling techniques, and encourage interaction through questions and discussions. Research findings must be carefully evaluated to identify insights that can meaningfully impact patient care or public health. Feasibility is a key consideration. We need to assess factors like cost, resources, and infrastructure required for implementation. Engaging stakeholders, from clinicians to policymakers, is crucial to ensure buy-in and address any concerns. Piloting new approaches, gathering feedback, and iterating on the process are essential to successfully translating research into practice. This multifaceted approach is the key to effectively bridging the gap between biomedical research and real-world impact. This video has explored the critical role of critical thinking and the scientific method in the field of biomedicine. These fundamental skills are essential for advancing medical knowledge, improving patient outcomes, and ensuring responsible and ethical research practices. Biomedicine requires a collaborative effort across disciplines, from clinicians and researchers to ethicists and policymakers. By working together and applying critical thinking, we can effectively tackle complex medical challenges and drive meaningful progress in healthcare. In the ever-evolving landscape of biomedicine, it is crucial to maintain a commitment to lifelong learning and staying up to date with the latest scientific evidence. By continuously evaluating and updating our knowledge, we can ensure that our practices and decision-making are truly evidence-based. These key takeaways underscore the importance of critical thinking, collaboration, and lifelong learning in the field of biomedicine.